Hello, marvelous people. In my previous videos, I referred to intrinsically disordered proteins, and I think that's probably a new term for many people, even scientists, because the way they function is different from some of the fundamental dogmas that has been set in biology over the past 100 years or so. So I think this presentation will elucidate some of those functions and bring home the idea that intrinsically disordered proteins are vital to biology and may be the next research frontier to curing many different diseases like prion disease. So this one is titled Chaos or Construction, Role of Glycobiology and Intrinsically Disordered Proteins in Prion Disease. Prion disease is also referred to as mad cow disease, or the scientific term is transmissible spongiform encephalopathy. But for practical purposes, I'm mainly going to refer to it as prion disease, or Kuru, which some ancient tribes used to call it by that name. So most proteins have a folded structure. All proteins are folded in three basic structures, either a beta turn, beta sheet, or alpha helix, but all of them can also be intrinsically disordered, which can be part of normal function, but also part of diseased function. So just to give you a cartoon of what that looks like, intrinsic disorder is many different conformations that the protein can be in. So it can bind different partners that are vital to its function, but also can lead to disease, which we'll look at in this presentation. So over 100 years ago, you had the Fore people in the East Highlands of Papua New Guinea, and they used to do ritualistic cannibalism to their elders after they died to honor them, so that they thought that the information from their elders would be directly passed down to them. And the women and children would eat most of the organs, and the men would eat the muscles. And they noticed that more often the women and children would come down with this disease that they called Kuru. And Kuru meant to tremble with fear. So what they were experiencing was a protein from the brain of their ancestors that was actually misfolded and causing some of their proteins to misfold in their brain, thus turning their brain into a sponge. And after this would happen, these people would refuse food and would eventually die from suicidal starvation. And these are all the other different names they have for the disease, because this can happen from the protein in the brain of many different animals, not just from humans. So the reason why this was really hard for science to understand at the time, and maybe still hard to understand and hard to do research on, is because it goes against the central dogma of molecular biology. So the central dogma states that information is passed from DNA to RNA to proteins. So the DNA can replicate itself and make more DNA. It can be transcribed in the RNA, and that RNA can be translated into proteins. And the central dogma thought that that's the only way information can be transferred. But the interesting thing is that this mutant form of the prion protein could cause other normal prion proteins to also get misfolded and disorganized and form these plaques in the brain that would eventually form a sponge-like structure in the brain, causing all kind of different behaviors to happen in that individual. So this suggests that the information is being passed from the mutant prion protein to normal prion proteins. And what would happen is the body would actually start making even more of those dysfunctional prion proteins. So this is an example of the proteins actually controlling the DNA. So the environment that all the cells are in is kind of dictating what the DNA is going to express. And we can see this in mothers that are obese or have some type of addiction like smoking cigarettes. They can pass that down to their children, and epigenetic mechanisms are given for why that happens. So you can think of it as the structure of these proteins outside the cell are going to bind to the receptors via glycoconjugates. So it's going to bind to the sugar residues, and that's going to activate certain genes because it's structurally going to make those genes available to be expressed. And then you're going to have an expression of certain genes that may be causing obesity or addiction to cigarettes, and that is going to get passed down to your children. So another way to visualize how this is happening to the protein, the prion protein, you can look at the normal prion protein. It has a few beta sheet structures, but it's mostly made out of alpha helix structures. And this is just a cartoon representation. This isn't exactly what it looks like. And then the disease prion protein has more beta sheets. So the disease prion protein, since it has more beta sheets, they can stack on each other and form these amyloid fibrils, which you can see right here. And the interesting thing is you can take some of these disease prion proteins, and if you put it in a test tube of normal prion proteins, it's actually going to cause them to form these thicker fibrils. And this is happening because of how the intrinsically disordered portions of the protein bind with itself and thus organize water, because all these proteins are mainly in water. And then it's going to organize the water and the protein in a way that's either very flexible and dynamic, which is how it's supposed to be, or more rigid, which is going to form plaques, which is seen in the disease prion protein. So in this way, we can think of biology as a complex adaptive system that not only does information flow from the DNA to the proteins, but it can also flow from the proteins back to DNA, which isn't really detailed by the central dogma. The central dogma just highlights one directional flow from DNA to proteins. So just because a protein is disorganized or intrinsically disordered doesn't mean that it's always going to be disease. If it's misfolded, then it could form disease, but some proteins, like the protein galectin-3, aren't misfolded. They're just intrinsically disordered. So the normal function of these proteins is to be a little disordered so that it can carry out that function. So you can see all right here, this is galectin-3, and even though it looks similar to the morphology of the prion protein plaques, it actually has normal function. So why would cells use this function for normal physiological mechanisms? Well, because you have things like cell adhesion and signal transduction of multiple receptors needed for immunity and development that need to occur and can utilize the aggregation, the dynamic aggregation of this liquid-liquid phase separation of these intrinsically disordered proteins. So these are some normal functions of intrinsically disordered proteins. And the prion protein also has normal function, even though it's intrinsically disordered. However, once it's diseased, that the conformations that that intrinsically disordered protein has, which it has many, is changed from some mostly alpha helical to mostly beta sheet structures, which ultimately leads to these amyloid plaques being made. So this is a better picture of the prion protein, which you can see is mostly beta sheets with a little bit intrinsic disorder in the form of beta sheets. But when the disease prion protein occurs, you have an increase of beta sheets, which is going to lead to an increase of these plaques being formed, and the brain forms a sponge morphology, so a sponge structure, which leads to dysfunctional behavior. So the progression of the disease is generally rapid and always fatal. However, it's not seen too much. It's only seen if you eat bad meat or if you cannibalize another human. So it's not really that prevalent, but there's no cure for it either. So science really hasn't caught up to this and figured out how we can prevent it. So statistically, as I said before, its prevalence is very low. It has 1 to about 1.5 cases per million, and this can happen not just from cannibalizing another human, but from eating bad meat from a cow or from some other animal. And there's no treatment for it as shown in the treatment strategies. All the medication is just to relieve symptoms associated with this disease. So future treatments, maybe they can use antibodies to block these proteins, or maybe antibodies that encourage the normal structure to take place. So the more alpha helical form of the prion protein can be promoted by antibodies. So maybe that's future ways that the pharmaceutical companies can go about treating this, but it's still up in the air. So I have a lot of conclusions, mainly that biology should be thought of as a complex adaptive system, and that prions epitomize the concept of complex adaptive systems, along with all kind of intrinsically disordered proteins like galectin-3, which plays a major role in breast cancer and other cancers. So understanding how these proteins function, I think, is vital for future research. So the take home of this is you are not defined by your genetics. So the environment that you put your cells in will actually dictate the genes that your DNA is going to express. And this doesn't just have to be an extreme case like prion proteins being dysregulated. It could be simpler cases like when a person smokes or when a person has too much fat in their diet. And so that can change the genes that are actually going to be expressed. So put yourselves in a good environment, all of your cells. Think about what you're putting into your body and how that might have an effect on your cells environment and also the DNA that's being expressed from those cells. And of course, cannibalizing human beings isn't good. You shouldn't eat human meat because it's going to lead to dysfunctional physiological conditions like transmissible spongiform encephalopathy. So I hope this was interesting, and have a wonderful day.